So, good evening to all the audience and speakers. We are here in the process of having a webinar series in conjunction with the National Medical Lab Professional Week celebration under the auspices of ICMLS with PGI team and me and other faculty members from different domains, different colleges and universities. So we have two very important talks today, one from Dr. Surendra Kumar Sharma, the other from Dr. Sumant Bera from ESIC PGIMSR JOCA. He is we have as moderator today Dr. Ashwinder Rana. He is a master's in pathology with specialization in cytopathology. I would request Dr. Ashwinder to be on video. Turn on your video, Ashwinder. Then he is a PhD in neuroscience. Currently, he is tutor, Department of Histopathology, PGMER Chandigarh. He has more than 29 years of experience. His master's, Dr. Rana has as an illustrious career, good record of publications. He is member of many national task force committees like Ministry of Health and Family Welfare, AIMS Bhubaneswar, Haryana and Punjab Board of Technical Education, QCI, that is Quality Council of India. A warm welcome to Dr. Ashwinder Rana. And as a moderator, I would request you to proceed with the proceedings. Thank you.
Thank you, sir, for a nice introduction. Thank you very much. And
my pleasure.
Very welcome to all the esteemed participants, faculty, and guests joining us today. So for their enriching webinar series, which we are organizing through the whole series, week, National Medical Lab Professional Week celebrations. So I have the pleasure of being the moderator for this session, today's session. So this week-long event is basically a tribute to the incredible contribution of medical lab professionals, basically, who work behind the scenes, but they have a very important but unnoticed role. And they are absolutely indispensable to patient care and medical diagnosis, right? So through this webinar series, we aim to highlight various key developments, share knowledge, and inspire learning across various disciplines in lab medicines. So before we begin, so I request all the participants, keep your microphones muted during the presentation. And secondly, if there are questions, so you feel free to post your questions in the chat box, so we can take them up during the question and answer session, right? So it gives my pleasure, it's my privilege to introduce my first speaker for today's session. That is Mr. Surendra Kumar Sharma. So Dr. Surendra Kumar Sharma, who is my colleague as well as a dear friend, is a distinguished professional in the field of medical lab sciences, who has done his PhD from PGIMER Chandigarh and has got nearly 25 years of rich experience, primarily at PGIMER Chandigarh, which is one of the premier medical institutes. His career spans from teaching, diagnostics, research, curriculum development, and institutional accreditation. He is an accredited assessor for paramedical and allied health institutions under the Quality Control of India, that's QCI, actively involved in evaluating and enhancing the standards of paramedical education across the country. So as a respected academician, he serves as a member of the Board of Studies for multiple universities, contributing to policy, academic planning, and course restructuring in allied health sciences. Dr. Sharma also has authored 14 research publications in reputed journals, which have nearly been cited over 800 times. So that is what underscores his scholarly impact and thought leadership in laboratory medicine. His expertise has been instrumental in shaping the medical lab technology curriculum for both the Haryana and Punjab governments, where he served as a subject expert for curriculum review and development committees. So with this brief introduction, I invite him just to speak on a very important topic, titled Technology Readiness Level for Commercialization of Lab Technologies and Technology Transfer. So I am honored to have him with us today. And Dr. Sharma, please, you can take from me now to have your talk. Please.
Very much, sir. Thank you for very elaborative introduction. It's my profound privilege to be part of this webinar series, and I'm so thankful to the team of ICMLS who has given me the opportunity to speak something about the topic which is very close to my heart. Can I share the slide, sir?
Yeah, surely.
The topic of today's presentation is technology readiness level for commercialization of lab technology and technology transfer. Throughout this topic, I'm going to cover about technology, what is technology readiness level, the process of innovation development that is divided from TRL1 till TRL9. I will talk about the BIRAC, which is Biotechnology Industry Research Assistant Council, which helps to provide the funding for development of such technology from labs to the industries. I will explain one example how we can develop the POCT kit, that is point of care testing kit. I'll talk about technology transfer process, its commercializing strategies, and finally I will talk about the patent filing. So I'll start with what is technology readiness level. So the technology readiness level, which is basically a measurement system that assess the maturity of technology as it progresses from research to commercialization. So we will evaluate from our research whether it is ready for the final commercialization. So the process development, the innovation development is having various processes from TRL1 till TRL9. In TRL1, we do the basic research. Then in the TRL2, we go for initial practical applications. Then in TRL3, we do the critical function or proof of concept is established. In TRL4, we do the lab testing and the validation of the prototype. In TRL5, we do the laboratory integrations and semi-integrated systems. In TRL6, we use to find out whether the prototype system is verified. In TRL7, we go for integrated pilot system demonstration. In TRL8, we do the system incorporated in commercialization design. And finally, TRL9, we do the whether our system is properly proven and it is ready for full commercialization deployment. So here, this is a graph which is showing from TRL1, TRL9, and it has the different levels. And here you can see that these are some red zones. Here you can see. So major research work used to be ended up to here. That is called the valley of death also. And our idea doesn't reach up to the level of full commercializations. So the level from TRL1 till TRL6 are very, very critical. And once our research is reached up to TRL6, then finally it may reach up to the final commercialization. So this is a funding agency which provide the assistance to different research organizations for development of such prototypes. And the thematic area of BIRAC is having the healthcare, which is having development of drugs, various biosensors, regenerative medicines, vaccines, and device and diagnostics. So our area comes here, device and diagnostics. So once we are planning to get some seed grant, we can apply through BIRAC and we can get the grants from this organization for maturations of our technology, which we are doing inside our laboratory. They do provide funding for the agriculturals, industrial biotechnological areas, as well as bioinformatics and softwares. So now I'll explain how we can go for development of point of care testing, which is relevant to medical lab technologists. Generally, the medical lab technologists used to do the different laboratory testings. So along with that, we do have our medical technologists are involved in the research area also. So we can find out which research may be reached up to the level of commercialization. We can go for development of any of POCT kits. We can develop some kind of sugar strips. We can develop for some urine proteins investigations. Even we can find out some staining techniques, whether they can be commercialized or not. So this is one of the example how we can go for the development of POCT kit. So it involves the different technological readiness levels. We need to assess them, whether this technology is ready for commercialization or not. So I'm going to cover about the roadmap of that. So POCT kit development is having integrated technicalities we need to find out. So how technically we are doing it, we need to go for the regulatory components also. We need to get the proper regulatory approvals before its commercialization. And we need to go for the commercialization milestones. So on the basis of first, that's the value of that. It's a lab-based research we do. And what is our objective during that from TRL1 to TRL3? That is, we need to have the concept as well as the feasibility assessment of the technology. So in TRL1-2, what we do the basic research. We used to identify the analyte, which we are supposed to identify, either the biomarkers or the different pathogens. In this area, we need to select the different detection methods. We can go for as per the research, as per the feasibility, as per the positive predicted values. We need to find out whether the lateral flow is working, or microfluidics is working, or electrochemical sensors. So we need to find out the area where we are supposed to do our basic research. Then we do the basic research for that. And finally, we go for the proof of concept. We used to see whether the lab settings is sensitive and as well as the specific for that particular testing, which we are supposed to go for it. In TRL3, we go for scaling of the prototype, lab scale of the prototype. We develop a functional prototype, either a paper-based strips or something handheld device, which we can see whether it is working properly or not. Then we go for the early validation of that. We see the different spiked samples, either whether our research is working with the bloods as well as saliva or the urines, and we see the ideal condition of that particular testings. In this TRL1 till TRL3, we are having several challenges. We need to ensure sufficient sensitivity as well as specificity. We need to choose the cost-effective materials for scalable production. And we need to assess the funding sources also. We can apply for different academic grants from BIRAC, from NIH, and early stage ventures capitals, or some charitable foundations is also used to provide some funding if our area of diagnosis is very novel. So in TRL4, once our technology is ready from TRL1 till TRL3, we reach at the level of TRL4 to 6. Here what we do the optimization of the techniques as well as assess the pre-commercialization validation. So basically it is a transition from lab to real world environment. What we do validate individual components, whatever we are using, either antibodies or sensors, in simulation real world conditions. Like we need to assess different temperatures, in humidity conditions, whether our device is working in different temperatures. Since we are having very worse temperatures in India, in winters it goes up to 2-3 degrees centigrade. In summers we reached up to 50. So we need to see whether the temperature is having some effect or not. We need to assess the integrated system of that. We assemble all the components in a user-friendly manner. Like we develop a plastic chip or a paper strip, we need to fix in the cartridge, as well as if we have developed some software connected with that particular sensors, whether the sensors are integrated with the software system or not, we need to assess that. Then finally, we do the bench testing. We compare performance against gold standard laboratory testings. As you know, we are having the different PCR ELISA. So if we are doing some POCT testing, qualitative testing, and we are getting some like urine strips, we are getting 2+ proteins, then we need to assess the real level of the particular proteins in that samples. So accordingly, we do the our bench testing. In technology readiness level 6, what we do the field testings in clinics. We used to provide free samples to different laboratories or different settings. We used to offer them free of cost and used to get their feedbacks. And once we are having a very positive feedback, then we can go for that next levels. If we get some negative feedbacks, we'll try to rectify that also. And accordingly, as per their feedbacks, we used to modify our technology, whatever we are using. One of my friend was have developed one OT thing. It was just a towel picker. So we took the different feedbacks from different organizations, and as per the PGI ENT OPD concern, they have said us like the the clamps are not properly fixed. So we worked on that. We talked to the company, and they fixed that, and then we applied for its patent as per the feedback we received. So the key challenges from TRL 4 to 6 is stability of reagents in uncontrolled environments. We used to minimize the false positive as well as negatives, and we need to find out because it requires a lot of funding. So we need to find out the suitable partners. We need to see the different companies who are involved in the already working, like we need to have some market for market surveys, as well as to give them in a market, as well as scalable production. So we need to sign some MOUs with them so that we can go for the large scale production of that POC ticket. So finally, once a technology reached up to the level of 6, so that is the value of that. So once a technology is reached up to the level of 6, we need to see the commercialization and scale up of that particular tip. Here we are talking about POCT. So we need to achieve regulatory approvals and market deployments. We need to go for clinical validations. We conduct the trials to meet regulatory standards. We do the clinical trials. FDA approvals we need to have. If we are developing POCT, then we need to get the CE IVD certifications, as well as we need to have some quality system implementations also. Like we need to have the MOU with the companies who are having the ISO 13485, which is for the manufacturing compliance. So once a company is having that compliance, that only we can have the MOU with that particular company, so that our technology can also be used in their facility, which is already having the integrated quality systems. So finally, at level 8, we go for pilot manufacturing. We establish the scalable productions. We see whether they are having the automated assemblies, how they are cutting our strips, how they are fixing it, and we need to submit the regulatory submissions. We need to file the IPR. If we are supposed to export it to USA, we need to go for FDA approval of 510. If we want to export it to European Union, we need to have their regulatory approvals like European Union, IVDR. And then finally, we launch the product at technology readiness level 9. We distribute throughout the healthcare networks, pharmacies, or direct to the customers, or we can sell our product through the different marketplaces. And we need to find out the close monitoring of that performance. We need to use to have their adverse events also. And once our product is successfully launched, then only we can go for final commercialization of that particular product. The key challenges in this area is we need to see the different regulatory pathways. Like as I already told you that India, we have the ISOs. In USA, we have that another approvals. In European Union, we have the different approvals. So we need to see our MOUs supposed to be signed with those manufacturers who are having the particular regulatory approvals. We need to see the competing with established diagnostics companies, because they are the old players in the market. They know how to cut the cost of the particular device. So that is also one of the challenge. And finally, we need to have the stakeholders like different manufacturers, regulatory consultants, distributors, and healthcare providers. We need to find out. Those will help our products to be distributed. So once we completed all the levels, there are certain critical success factors which we are supposed to be kept in mind. We need to see that our product need to be aligned the design and regulatory requirement as per either FDA, EU, or ISO. We need to see their quality controls. Since it is POC ticket, so in that area, we need to go for ISO 15189 for medical laboratories, whether it should be compliance of that also. There are various market differentiations. We need to see the target unmet needs, that is rapid sepsis detections at home, STI testings. We can develop the POC ticket for these particular areas, which till date has not been developed. We can detect some sort of rapid sepsis detection kit. A person can detect that sepsis at their own home. We can go for the cloud connectivity of data analysis, the trend monitoring. We can go for it. These are areas which till date is untouched and the research is still going on. We need to see the cost efficacies because the rates of different materials are different. Like nitrocellulose for lateral flow, we can use the simple paper sticks also, but stability is testing and the production methods will vary the cost of that particular instruments. And if we go for the larger scale productions from different companies from South Asia, we can get the lower cost manufacturing as compared to if we sign a MOU with any European company or American companies, then we are going to get the higher cost of our product. In conclusion, development of a POC ticket requires parallel progress in technical validation, regulatory compliance, as well as marketing strategies. And once a person is aligned all TRL milestone with different stakeholders, they know the funding mechanism. Lab can bridge the valley of death, which I already told, that is from TRL1 till TRL6. So once a laboratory is covered that valley of death, then we can go for delivering a impactful diagnostics to the end users. So now I'll talk about that, how we can go for technology transfer process. So basically technology transfer process, the gap between research institutions and industry partners. So we need, the key areas are, first of all, assessment, then IP protection, then partnership development, then finally commercialization support. First, we'll evaluate the technologies levels, its market potential, and IP landscape. We identify the potential industry partners as well as licenses. We go for IP protection of our product. We file the patents. We get the copyrights to protect our innovations. And at that time, we can negotiate for licensing agreement with different agencies. And if we will not sign an agreement at this level, IP protection level, so the larger players can hamper your rights. We need to have a proper partnership development documents. In that, we collaborate with different companies for further development of the product, because a research laboratory cannot scale up their production in the research setting. We need to have a partnership with the already established companies. Those are having different compliances for production of different type of products. So once we identify which POC ticket or which type of sensors we are developing or any dye we are going to establish. So as per the market survey, we used to find out the tentative partners of our product. Then we need to have a proper partnership agreement with them. And then we get the commercialization support. These type of companies used to provide different technical assistance for the scale-up production of our products. They facilitate access to funding or investors. Even ICMR is having a separate unit which provide such sort of helps to different research laboratories, even a private organization, private universities also. If they are developing some sort of innovations, how you can protect your innovation, how they can try for their TRL level assessments, and finally scale up of your product with different companies. They do have their specialized consultancies through ICMR. So these are the basically stakeholders. In a product development, we used to have researchers, those who develop the technology and provide technical assistance. We used to have another stakeholder, which is technology transfer officers. Those used to manage IP, negotiate licenses, and facilitate partnerships. We used to have another stakeholder, which is industry, which provide the funding for manufacturing capabilities and market assessments. And finally, a regulatory body, which ensures the compliance with different standards. About the commercialization strategies, first of all, we need to have a proper license of that particular technology for development as well as the distribution of that product. If a company is having the universal license, so it is going to be easier for us to export our product to any of the country in the world. Spin-off creations, we need to form a startup to commercialize the technology. Even nowadays, Government of India is allowed, to create a non-for-profit or a company inside government organizations, where a team of researchers can create a startup company which can go for development of any of such type of products, which is now allowed in government institutions also. And we also can go for joint venture, where this type of research group as well as the industry partners can joint venturely product a product for commercialization strategies. Like a private university and a medical device company can make a new implant, or they can make a new device, or they can make some technology which through AI, through mobile, can help with the diagnosis of the patients, early diagnosis of patients. And then finally, I'll talk briefly about the preparation of patent applications. Because at the end, if we develop a technology which is having the scalable potentials, so first of all, we need to have the patent for that particular technology. And here, what we do, first we do patent search, we file a patent application, and finally we get a patent. And here is a website. You can take the picture of this, and by clicking of this website, you go straight away to this page. This is a Indian Patent Advanced Search System. Here, suppose if we are doing some research for developing a POCT kit through lateral flow or through nitrocellulose strips, if the technology has already been established, already been published, have the patent on some other name, then there is no logic to go for it. So before to start any of your area of interest, you need to find out whether this is already a patented area or not, or there should be some variations which the technology you are going to get patented. So through this website, you just go for a straight search and you will get the different patents which we are having. Even if you put up your patent number, like once you will see any of the product in your hand and you see the patent number of that. Suppose we are going to develop a POCT kit and that is having some patent number. We can put patent number over here and can identify whether it is having a genuine patent or not. So different organizations like this is Ministry of Education's website, which provide even funding for applying of patents. Here, different schools or different university students, they can also apply for patents, and they used to provide that amount which is required for filing of patent through this Ministry of Education's initiative. So through Kapila Yojana, we can get that funding from this agency, 2,000 to 3,000, whatever the amount is required for filing of patent. We used to get it from this website also. So we can encourage our students, if they are having some innovations, go for searching whether the innovation is already having a patent or not. If it is a unique idea and having some scalable opportunities, then we can tell them to apply for this kind of different government institution for filing of patent. So this is a program for all higher education institution. We get the 100% patent. They used to provide 100 patents per year, per institution. So even a university or a single institution can apply, 100 of the students can apply in a single year for 100% of funding for patent applications. So this kind of government initiative helps our students to file their innovations for the patents. Even the patents also can be sold to any of the the established companies where they used to have their own protections. Like they can have, we all can have our percentage in the once it is a scalable production. So this initiative provides the IPR awareness. They provide the funding assistance of 5,600 INR per application for patent filing. So this is a cost which is required for the IPR filing. So they used to provide 100% funding, 100 students per year per university or per institution. So here, this is the announcement by our health minister, where they have encouraged the indigenous innovations and advanced research for our nations. And nowadays, a lot of budding research institutions are giving the applying for IPR protection and different type of filings. So in conclusion, the process of innovation development guided by TRLs requires a coordinated effort between researchers, technology transfer officers, and industry partners. Because in isolation, a single person can't do anything because it's a huge work from research till the industry level. So we need to have a proper team of that. And if we align all these technical progresses with commercialization strategies, the technology transfer mechanism, lab-based innovations can successfully transition to market-ready solutions. So this is the area. And here, the picture I am sharing with you, it's a picture of last year after NMLPW. I have been given an opportunity by ICMLS to reach Incheon, Korea, where we have signed a MOU for Indian medical technologists to go there in Korea and to do free of cost training in their institutions for how these laboratories are working, how they are getting their technology to be regulated. So ICMLS has given such kind of opportunity to their members also. Thank you very much. If any questions, I'll be happy to give the answers. Over to you, Dr. Ashwin.
Thank you, Dr. Surinder, for such an informative talk. That's very relevant to the time. And so these TRLs, technology readiness levels, provide a structured roadmap to move, lab innovations towards market-ready solutions. As you said, successful commercialization needs these lab technologies. They depend on the technical maturity and also the strategic technology transfer, regulatory alignment, and industry collaboration. So all these combined together, we can achieve this. Thank you. Thanks, Dr. Surinder. If we don't have any questions, we can move to the second one. Right. So in our second talk, we have got another eminent speaker. I'm privileged to introduce the second speaker of today. He is Dr. Sumanta Bera. He's an expert in the field of medical lab sciences with a remarkable career in spanning nearly over 3 decades. Dr. Bera currently serves as a scientific officer and in charge vice principal at the ESIC College of Allied and Health Professional, ESI PGI MSR, Medical College and Hospital, Joka, Kolkata. A highly qualified professional, he holds a PhD degree and has got a gold medal in MSc while doing MSc in Biomedical Lab Sciences and Management. And he has also got a postgraduate diploma in lab technology from the various, from prestigious school of tropical medicine, which is under the Calcutta University. Throughout his career, Dr. Bera has led advancement in medical lab technology with the focus on andrology, diabetes markers, and thalassemia screening using HPLC, backed by 7 years of hands-on expertise. He is an integral part of NABL and NABH accreditation process, a certified ISO internal auditor, and a committed troubleshooter for quality lab operations. In addition to his technical excellence, Dr. Bera is deeply involved in teaching and academic leadership, student mentoring, and patient care counseling, reflecting his unwavering dedication to both healthcare delivery and human service. He also has a role as an innovative social worker and advocate for patient-centric care, which exemplifies the very spirit of this National Medical Lab Professional Week as well. So with this, I'm honored to welcome Mr. Bera to speak on a topic which is very relevant to the time, that is diabetes-induced male infertility, an approach through diabetic profile and andrological biosensors. So we are honored to have Dr. Bera. So Dr. Bera, please continue with your talk.
Good evening. Good evening to all.
Thank you.
And thank you, Dr. Asbinder Raina, sir.
Thank you.
Very good evening. Good evening to all, and all the delegates, and very warm welcome to our National Medical Laboratory Professional Week 2025 webinar. And thanks to our ICMLS, they have given me the opportunity to present my research work. And at first, I want to acknowledge to my Professor Devidas Ghosh, who is guide, and he was the philosopher of my career. And today my topics is diabetes-induced male infertility and approach to the diabetic profile and andrological biosensor. And I want to place the present the screen. Yes. Can I want to share the screen?
Just a minute.
Yes.
Give me a minute. Kaptanji,
Okay.
you have to assign him co-host.
Okay, sir. Just a minute. I am trying.
Actually, at this time, I can tell what is diabetes. Actually, diabetes is the disorder of carbohydrate, fat, and protein metabolism, where insulin deficiency or inefficiency will be there, associated complication, neuropathy.
Yeah. You are co-host. Dr. Bera, you are co-host now. You can share your screen.
Yes. Thank you, sir. Yes. Is it visible to all?
Yes.
This is my topic, diabetes-induced male infertility and approach to diabetic profile and andrological biosensor. Actually, in simplest, in the diabetes word, it indicates the disorder of carbohydrate, fat, and protein metabolism, where insulin deficiency or inefficiency will be there, associated with complication of neuropathy, nephropathy, and microvascular disease, where blood glucose level, C-peptide, insulin, glycated hemoglobin will be elevated. And as a symptom, it will be expressed excessive thirstiness, hungerness, urination, that is polydipsia, polyphagia, and polyuria. And it is the third leading cause of death, where the erectile dysfunction may be observed, and it's a syndrome. And the male fertility when a couple is allowed to mate without the help of any contraceptive practices for 1 year, but there is no issue, then it referred as infertile couple as per WHO guideline. If the male partner is responsible for infertility and that is due to diabetes, then the condition is known as diabetes-induced male infertility. The international scenario, if we see that World Health Organization updated on 14th November 2024, the number of people living with diabetes rose from 200 million in 1990 to 830 billion in 2022. Prevalence has been rising more rapidly in low and middle income countries than the high income countries. More than half of people living with diabetes did not take medication for their diabetes in 2022. Diabetes treatment coverage was lowest in low and middle income countries. If you see the national scenario, India is among the countries with the highest diabetes prevalence, ranking second after China. In 2021, it was estimated that 77 million people in India had diabetes. And the number of people with the diabetes in India is projected to increase 124.9 million by 2045. Though the regional variation may be observed, the burden of diabetes varies across the different states in India, with southern and western region experiencing higher prevalence compared to the northern and northeastern region. And this is the recent study in 2022, 40% adult age group of 18 and older were living with diabetes, an increase from 7% in 1990. More than half, that is 59% of adult age group 30 and over living with diabetes were not taking medication from their diabetes in 2020. In 2021, the diabetes was the direct cause of 1.6 million deaths, and 47% of all deaths due to diabetes occurred before the age of 70. Another 530,000 kidney disease deaths were caused by diabetes, and high blood glucose caused around the 11% of the cardiovascular deaths. Several complications, basically we can divide into macrovascular and microvascular. In microvascular, what problem related to our topics is sexual dysfunction. To search out whether the patient is diabetic or not, and then the quality of sperm and sperm function test in the diabetic patient, to find out whether the diabetic mellitus-induced male infertility is linked to oxidative stress or of spermatozoa, to search out whether the diabetic patient is hyperlipidemic and loss of libido or not, to evaluate testosterone, to focus on tuning system of hypothalamico-hypophyseal testicular axis. That the selection criteria of the diabetic patient was suffering from diabetes for 4 to 5 years at least, and fasting blood glucose level 150 to 170, and age limit 30 to 50 years. Similarly, the matched normoglycemic patient selection was same, age group 30 to 50 years, normoglycemic still now, and same community and religion, and food style and work style more or less same with the diabetic group. This was my experimental design of the human object with 30 to 50 years. I have divided into two groups, diabetic group and non-diabetic group, that is control group. We have taken blood sample and seminal fluid. From the blood sample, we have estimated fasting and postprandial blood glucose, glycated hemoglobin, C-peptide, serum lipid profile, serum testosterone, fasting and postprandial insulin level. And from the seminal fluid, we measure the volume, see the viscosity, motility, total count, viability, morphology, and density. From the seminal plasma and pellet, we estimated catalase, peroxidase, superoxide dismutase, TBRS, conjugated dye. And from seminal plasma, additionally, we estimated the fructose level. Semen volumes, sperm count, sperm motility, sperm morphology, and progressive sperm motility we have done, and the sperm viability was estimated by Yosin Nagdoshin. Catalase activity by H2T, substrate catalase activity was measured, reading at 240 nanometer. Peroxidase activity of sperm pellet and seminal plasma in presence of guaiacol peroxidase activity by recording OD at 436 nanometer. And SOD also estimated by the percentage of inhibition in the pyrogallol auto-oxidation by SOD at 420 nanometer. CD marker level of from sperm pellet and seminal plasma was extracted using chloroform methanol mixture, and lipid residue was dissolved in cyclohexane followed by absorbance measurement at 233 nanometer. TBRS level activity of sperm pellet and seminal plasma using TBA-TCA mixture with sperm pellet seminal plasma quantity of TBRs was monitored by recording the absorbance at 535 nanometer. Fructose level seminal of spiral plasma also used for the purpose of using standards concentration of the fructose was measured, noting the absorbance at 470 nanometer. Blood glucose level normally conformed method by EMERC, and glycosylated hemoglobin column ion exchange method Crest biostat kit. Serum testosterone, C-peptide, insulin level, cholesterol, triglyceride also by randomization. The sample was collected and in a wide mouth sterile polystyrene graduated container separately. Color, odor, pH, semen sample of the both groups were studied. Abnormal semen from these parameters was discarded. Volume measured after natural coagulation and liquefaction process. Sperm count, sperm viability, sperm motility, acrosome function, and HOS test were measured. Antioxidant enzyme activities were noted with quantification of product of free radicals. That's why the patient consent was also taken. What result we have seen and the discussion was done. In case of control group, the fasting blood sugar, blood glucose level was better than the diabetic patient. And in diabetic patient, PP sugar and fasting sugar was elevated. Effect of diabetic mellitus on glycated hemoglobin, the serum insulin C-peptide level of diabetic patient in compared to the matched control group. Also, it has been observed that in diabetic patient, this 2 level is increased. On the same way, we estimated the fructose level. We have seen in diabetic group, this level is increased in compared to the control group. In diabetic group, the catalase is decreased, and in compared to the control group. Peroxidase also, that is also decreased in the diabetic group in compared to the control. In diabetic group, superoxide dismutase is also decreased. On the other way, the CD marker conjugated dye-in, it increased in diabetic case. It has also estimated from the sperm pellet as well as seminal plasma. TBRS increased in diabetic group, and we have seen that testosterone level in diabetic group is remarkably decreased. And chronic, in case of semen volume, it has been observed that volume is diminished in case of a chronic diabetic patient. And its sperm concentration, motile sperm concentration, and functional sperm concentration is also diminished in case of diabetic case. And the sperm function test for estimation, the sperm viability of diabetic patient and sperm viability of a control have been estimated by Yosin-Nagdoshin method. We can see that in control group, alive spermatozoa around 70% and dead spermatozoa is 29%. We can see that dead spermatozoa is colored as deep red and alive spermatozoa is dotted as white color. In diabetic group, that alive sperma is around 35% and dead sperma is 65%. It is hypoosmotic swelling test. It has been done also in case of diabetic patient as well as control group. You can see that sperm swollen tail, that is in case of control group, that is 70%. Sperm with non-swollen group, in diabetic case, that is 35%. So due to the membrane integrity deficiency, in case of diabetic patient, its swollen tail has been diminished and non-swollen tail is increased. And other test, nuclear chromatin decondensation test, that is also observed that sperm with decondensed head in case of control group is 70%, in case of diabetic patient that is 35%, and sperm with condensed head is 29%, and diabetic patient 65%. And acrosomal status also we can see that sperm with hallux formation, we can see it as a control patient, we are getting it 80%. In diabetic patient, that is 35%. Sperm without hallux is 20%, and diabetic patient 65%. High level of lipid profile, which can cause the impotence, loss of libido, and act as unfavorable on erectile dysfunction or erection. In this case, you can see that in diabetic group, the serum cholesterol is high and triglyceride also increased. So you can see that subject patient who are suffering from diabetes, 3 bar diagrams support and confirm about the subject is suffering from diabetes. What I have already said. And the level of fructose of seminal plasma due to non-utilization of the low sperm count, that is oligospermia, leads to chances of male infertility. And low level of antioxidative enzymes like catalase, peroxidase, superoxide dismutase, and high level of free radicals and products, that is CD and DBRS, support the excessive oxidative stress generation in sperm, and plasma may be associated with the serious defect in sperm morphology and seminal quality and function. Excessive oxidative stress may be associated with the serious defect in semen characteristics, such as severe oligospermia. The generation of reactive oxygen species, that is ROS, in the male reproductive system has become a real concern because of their potential toxic effects at high level on sperm quality and function. Decreased sperm motility, abnormal morphology, and functional and motile sperm are abnormal with decreased level of sperm mobility, maybe due to the impaired testosterone production from Leydig cell function, and oxidative stress generation in sperm and seminal plasma leads to reduced fertility. High level of lipid profile, which can cause impotency and loss of libido and acts as a unfavorable on erection. So diabetes and male infertility and investigative conclusion, low insulin level or insulin resistance, and that which happens, glucose insulin axis alteration, altered tuning system of the pituitary testicular axis. That's why LH and FSH becomes low, giant cell formation, and that ultimately leads to male infertility. And the low testicular androgenesis, low testicular testosterone, axillary sex glands abnormalities, and erectile dysfunction will happen. So diabetes mellitus affect the male reproduction, low sexual desirenousness, oligospermia, low sperm viability, motility, oxidative injury of germ cell, testicular oxidative stress, diminution in the testicular androgenesis, and diminution in the plasma gonadotrophin. So diabetes can be prevented by lifestyle management, but diabetes complication cannot be prevented if diabetes is not controlled. Thank you.
Thanks a lot, Dr. Samanta, for this. I think this is a research paper we have presented.
Yes, sir.
Yeah, that's a very nice study because these days there's a growing concern of this, diabetes related infertility. So just I want to see whether this has been published or this is.
Yes.
This has been already published.
Yes, published.
Okay.
Actually,
Right.
it has been published in 2017.
2017. Okay. Right. Any questions? So we can take that.
Yes, sir.
If we have no questions, so we come to the end of this session, and I am thankful to all the participants, speakers, faculty who have joined today for this webinar series. And I will fail in my duties to have a special mention of Dr. Shalini Bajaj from SGGPS College of Pharmacy and their faculty and students for the enthusiasm they have shown by participating in this webinar series. I thank you all, and that is it for today. So tomorrow we'll meet again at 6 p.m. for another series of lectures. So then I hand over to Dr. C.P. Pandey sir for closing the session. Thank you.
Thank you so much. It was really a wonderful presentation, and this is the fourth day we are into the celebrations of NMLPW-2025 under ICMLS, and all the lectures have been up to the mark. They were quite educative. As you said, we are very thankful to the faculty, Dr. Shalini Bajaj, and her students who are regularly attending these webinars. They would definitely be benefited out of it. Thank you, Dr. Sumant Bera. Thank you, Dr. Surendra Sharma, and all those who have joined. So, bye-bye for now. Tomorrow we'll meet again at 6 with 2 talks, one from PGI faculty and the other one from somewhere. I forgot who is that person. Maybe she is from Gujarat, Anand, right? So those will also be quite educated and informative. Thank you, Dr. Sumant Bera, once again, right?
Thank you, Dr. Pandey and Dr.
Yeah,
sir. Thank you, sir. Okay.
Namaskar.
Good night, sir.
I'll be failing in my duties if I don't mention my friend Dr. Ashwinder Rana. Thank you for moderating the session. Okay then. Bye-bye. Bye.
Okay.
See you.